Good morning, everybody. So we have today with us a member of our own team, and it's my pleasure to introduce him. Sergi Taboada is a member of our department, Evolutionary Biology and Biodiversity, where I'm the head. And he's a Ramón Cajal with us since recent. So he started this year. He works on evolutionary biology, marine biology, and marine invertebrates. If you look at his CV, the word that shows up most often is connectivity. So he works on connectivity of populations with genetics, and mostly genomic tools nowadays, phylogenetics, phylogenomics, population genomics, always on marine organisms, mostly invertebrates. So he started his career in Barcelona, where he got his degree and PhD, and then moved as a postdoc to London, to the Natural History Museum, where he stayed for about 3 years. And then he came to Madrid and did a circle through all universities in Madrid. He had Juan de la Cierva in Universidad Autónoma. Then he moved to Universidad de Alcalá with another postdoc grant. Then he moved to Complutense as assistant professor. And as I'm saying, very recently he joined us with Ramón Cajal. Over his career, he had 9 fellowships. He has been very active. 9 fellowships. He has done 65 presentations in conferences. He has done a lot of teaching, tons of hours. He has 3 PhD students, 1 finished, 2 in course. He has supervised a postdoc. He has supervised over 25 master theses and similar number of bachelor degrees. So you see he has been pretty active scientist. He has over 70 papers published in relevant journals, and he has now 3 running grants, one of the national, so Spanish grant, another Biodiversity, and participates in an EU project. So you see very active scientist, and yeah, sorry, the floor is yours.
Can you hear me? Yeah. So thanks a lot, Marta, for the introduction. Voy a dar la charla en catalán porque como soy catalán, es broma. Just a joke. It'll be in English. It's my pleasure to be here to present part of the work that I'm doing in here, the work that I've been doing recently within the past few years, related to conservation, genomics in marine ecosystems. So this talk is going to be kind of an overview of what I've done, what we'll be doing in the near future, and what we're currently doing right now. So if we Google marine conservation, you probably wouldn't be surprised to get different web pages of NGOs, mostly NGOs, where you find different flag species, emblematic species, penguins, whales, polar bears, turtles, species that attract the attention of the people for conservation. And in some cases, you can also find a few marine invertebrates like corals. So these emblematic species are capturing our attention due to the problematic related to climate change in the ocean that are causing shifts in species community structure, shifts in species biogeography, ocean acidification, coral bleaching, and also causing disease and death, which is increasing extinction risks for all these species and the communities where they live. But not only these species are going to be affected, and this is a spoiler for the rest of the presentation, because marine invertebrates also matter for conservation, and this is what this talk is going to be about, with examples like sponge rounds, as you will see later in the presentation, which play a crucial role in the ecosystems where they are found, or also marine worms or brittle stars and emertins that also are useful for conservation purposes, as you will see later in the presentation. But just before conservation, we need to know what's in there. We need to know, we need to describe, we need to delineate species. We need to establish which are the units of conservation. And this is why we I consider myself partly as a taxonomist. We are necessary. And this is especially true for marine habitats where biodiversity efforts to describe species has been, in comparison with terrestrial habitats, is less. And where also there's a high prevalence of cryptic species. So to solve this problem, we have to apply new tools. It's not just describing the morphology of the species. In order to describe these units of conservation, we need to apply integrative taxonomy by increasing the information about genetics, the distribution of the species, the ecology of the species, morphometrics, behavior. And this is one of the aspects that in which I've been devoting part of my career, describing new taxa in the areas where I've been lucky enough to work. So far, 20 new species, mainly from marine worms, annelids, my favorite group, but also from emertins, sponges, and other organisms belonging to other phyla. And just to illustrate what we do in our lab, I'm just showing here an example of, this is a carnivorous sponge, like 40, 50 centimeters long in the Cantabrian Sea at around 1,000 meters depth. This is a gorgeous species. In the Cantabrian Sea, as I mentioned, it's Chondrocladia robervalardi, but I'm not interested in the sponge. I was interested at the very beginning, but since I found some worms living, well, you will see it now in a minute, but the worms are in here, living together with this sponge. And what we did with this worm is we described, it's just to illustrate what we do in our lab. We placed this worm within its phylogenetic context using different genetic markers. So this helped us to put into context this species, but also to name this species. This species is named Neopolinoi chondrocladiae. But also really we're interested in describing more in detail this species and their relationship with this Chondrocladia robervalardi, with this carnivorous sponge. And this is what we did with this microcomputed tomography, which allowed us to identify how the sponge and the worm are behaving. And we identified that it's actually the sponge that builds galleries that accommodate the worm. So the worm is living there because the sponge wants the worm to be there. It's a non-destructive analysis, which is great. We were using collection material for this, in this case. But not only this, we also were describing some features about the worm, and we identified, using SEM, hooked kitty that facilitates the anchorage and navigation along the host, which is indicating that it lives there because, it's an adaptation of living there. But also we were interested in some of the structures that these organisms, these worms, they do have. Those are elytra, as kind of scales, and we were studying the histology of this elytra, and we identified photocytes, which are actually related to producing bioluminescence, something that was also corroborated by our confocal analysis on these scales. And we suggested that the worm is producing bioluminescence to attract more prey for both the sponge and the worm. So this is just to show you, with this all the information, we inferred that this symbiotic relationship, this unique symbiotic relationship between the sponge and the worm is mutualistic. This is just to illustrate what we do in our lab in order to describe which are the units of conservation, which is the information that we need to have if we want to do conservation, because the next step is studying the connectivity along the different populations. And this is, again, quite difficult in marine organisms, particularly for the biological features that they normally have. And I will show you in a minute. Marine organisms, they normally have larvae that they use for dispersal, that they can be either lecitotrophic. This is the most common lecitotrophic, the most common larvae for sponges. they don't fit in the water column, so they can last in the water column for a few weeks. But they can also, there's also organisms with plantotrophic larvae. They can feed in the water column, and they can stay in the water column for even months. In general, marine organisms, they also have high fecundity. They have big populations sometimes, very wide distributions, which makes really important to make marine protected networks that allow connectivity, sharing connectivity and genetic diversity among different populations that go widespread along the distribution of the species through corridors or stepping stones populations. But this is just, what I just mentioned is just related to the biological features of the species. But there's also, since I mentioned that most of the species are going to have larvae, dispersal of these larvae is going to be mainly passive and mainly using these oceanographic, predominant oceanographic currents. So for conservation, we need to implement not only this biological information, but also the abiotic information of currents like the ones that I'm showing here, in here. So it's crucial to integrate this information, as you will see later also. So maintaining connectivity involves creating ecologically connected and functional networks, as I said, with corridors or stepping stone areas that facilitate the movements of individuals or genes. And how can we evaluate connectivity in the wild? This is what we do now. It's by the use of genetic markers. Which genetic markers? During the talk, I'm going to be talking about traditional markers, which is a small portion of the genome, either mitochondrial or nuclear, that can be used to build these haplotype networks. These haplotype networks, every circle corresponds to a single sequence or haplotype, and the different colors in this case correspond to different areas. So in this case, we show a clear differentiation between this red area with respect to the other areas. But there's also other ways to start and to use these genetic markers or other genetic markers, such as microsatellites. We've used that in the past, and they will appear in this talk. And also SNPs, single nucleotide polymorphisms, which are the ones that we are using currently in our lab through this protocol, DD-RADSIC protocol. So with all this information, we can build this kind of plots, which is this structure plot. Each bar is an individual. Different populations are depicted in here, and the different colors, they correspond to different genetic clusters that the program infers, and the analysis infer. So this genetic information can be used, of course, to detect, which is the genetic connectivity through the migration patterns, and how this connectivity ensures genetic diversity being shared. But also we can quantify genetic diversity, which is a parameter which is supposed to be directly linked to resilience. So low genetic diversity can lead to a reduced adaptive capacity if in a changing environment in where all these organisms are living. So in this talk, I'm going to be talking about three main regions in where I'm going to be showing the problematic of this region, or the human threats for the different regions, and the way we have tackled the different our approaches for conservation. We'll be talking about Antarctica, Clarion-Clipperton Fracture Zone, and also in the North Atlantic, with the aim to answer these three fundamental questions. So what is the genetic diversity of the populations or the species level, which is, as I said, related to resilience? How connected or isolated populations are, which is crucial to define different marine protected areas, for instance, so for the design of the marine protected areas, and which are the physical or behavioral factors driving this connectivity. And my focus will be mainly put into oceanographic models that we have developed to explain that. So we'll start with the Antarctic Peninsula. So you probably think about the Southern Ocean. One of the first pictures that will appear in your mind will be icebergs. Other pictures will be penguins or seals or whales. Probably you will not imagine myself there. I do imagine myself there because I've been there. Yeah, those are the flag species, right? But those are not the ones that I'm interested in. We are not interested in. We are interested in what lies below the surface. And I'm going to show you this video. Scuba diving in Deception Island. This is one of the worlds that we were visiting routinely in there. And as you can see, there's gorgeous life in there. There's many different species, huge species of different sponges, also echinoderms and other marine invertebrates associated. So there's a lot of life in there. Also humans trying to get samples into the right place. This is what we've normally done in shallow waters because we've collected many samples in there, but we've also used cutting-edge techniques such as turning rocks in there in the intertidal so that to find little creatures that I will show you later. So let's talk about a little bit about the Southern Ocean. The Southern Ocean is an isolated ocean, and it was separated from the rest of the continents like about 20 to 30 million years ago. And they are physically separated from the rest of water masses thanks to the polar front, which is different temperature from the rest of the water masses, and also thanks to the Antarctic circumpolar current. Although it's isolated, it's also experiencing one of the highest increases of seawater temperature worldwide. This is the air temperature. We're all experiencing this ourselves. It's a picture from 2022, but I would like you to focus on that. This is the Antarctic Peninsula. This is also experiencing a huge increase of temperature. But this is also translated into the seawater temperature. As you can see here, the increase of the seawater temperature, which will be affecting directly to shallow water species. So in a study that we did in 2015, we wanted to assess how much we knew about shallow water specimens, shallow water species in the Southern Ocean in terms of genetic connectivity. And what we found is that most of the studies were conducted for deep water specimens, and few studies were done at the regional scale, and particularly very few on the Western Antarctic Peninsula, which is one of the areas that, as I mentioned before, that's going to be being affected by the increase of the seawater temperature. So there was a need to find shallow water model species to study connectivity and phylogeography patterns at the regional scale, in a particular with a particular focus on the Western Antarctic Peninsula. And this is what my former student, PhD student Carlos Deva, in his PhD was devoted to mainly and try to answer this. He had enough of cold temperatures. He's now in Guam. He's now enjoying himself in another weather. But apart from, well, he did many good contributions on science related to connectivity, but probably one of the most important ones, and the one that I will be highlighting now, is this last one that was recently published. I will explain in a minute about it. So what we were interested was in studying which was the degree of connectivity between different areas. These green areas are marine protected areas. This is the South Georgia Marine Protected Area. It's a huge area. This is a South Orkney Marine Protected Area. And there's also this D1 MIP proposed Marine Protected Area, which is what's proposed by a Commission for Conservation on the CAMELAR. So the idea for this study was to try to answer this question about, is this domain 1 MIP needed to create a huge marine protected area network? To do that, we collected through many different Antarctic cruises a huge amount of specimens with different reproductive strategies, plantotrophic, lexitotrophic, or brooders, with a priori different dispersal abilities, high, medium to low dispersal abilities. And we also combined different sources of genetic approaches, like using CO1, a traditional marker, and also DD-RADTIC. And well, this is the start. I mean, the summary of what we did, or what we found with both the CO1 and SNPs agreeing in that there's a clear separation between South Georgia, which is this area to the north, and this D1 MIP, as you can see here. So you see this red area, which is the South Georgia, and the rest, which is the D1 MIP, which corresponds to the Western Antarctic Peninsula. The same happened for the SNPs, where we found a clear separation, clear genetic separation in between them. Not only that, but we also found that the organisms in the Western Antarctic Peninsula had high gene flow among the different populations found in this area. And at least for one of the species that we studied, we found that there's the Sodornis, this area in between these two big areas, plays a stepping stone role, between South Georgia and D1 MIP. So you can find there organisms with affinities to both the South Georgia and D1 MIP. So all these results point to support at least the implementation that, we are pushing hard for this implementation of a new Antarctic MPA, this D1 MIP, which hopefully in the near future will translate into something real. So let's move now to the Clarion-Clipperton Fracture Zone. Probably most of you are not familiar with that, so I'm going to also briefly explain. This is what you see to the north, to the south, to the east, and to the 360 degrees, because it's in the middle of the Pacific. But again, I'm not interested in what happens on the surface, but what happens at 4,000 meters depth. This is totally different aspect, as you can see from the last one. This is clay that you see here, and these small rocks, these are manganese nodules, which is the main reason we are there to do our science, and the main reason we are trying to describe what's going on in there. From time to time, you see some very big organisms. It doesn't mean that it's not a really diverse area, because there's a lot of really tiny organisms that you will see in a minute. This is just a closer look to these manganese nodules. This is me, that go from few centimeters to lots of centimeters. And I just brought some manganese nodules, which are here, in case you want to have a closer look, and you can also touch them. We're not, no cobramos nada. So we are interested in this, and the reason why we're interested in these nodules is because they harbor a lot of minerals that are interesting for humans. They are called ferromanganese nodules because they have manganese, iron, but they also have nickel, copper, and other rare elements that are potentially being used, could potentially be used by humans. This is the main reason research is being there, conducted being there. What's the problem? The problem is that, as you can imagine, extraction of these nodules will be very aggressive. It will be removing nodules, but not only that, it will be producing sediment plumes. This clay that I showed you before will produce sediment plumes that will last for many months in the water column. And also, these nodules are extremely low recovery. They grow on a rate of 1 centimeter per million year. So there is a need to regulate this extraction and to provide environmental assessments, and this is why we did our study in there. So this is just to have an idea of how big the area is. It's 12 times the surface of Spain. It's a huge area. And the polygons in different colors that you see in here, those are the claim mined areas. It means that some countries have claimed to mine there in the near future, probably in a few years or even less. And the squares that you see here, the green squares that you see here, are areas of particular environmental interest, or APEIs, that were designed by the International Seabed Authority a priori to safeguard biodiversity of ecosystem function. And our idea was to test whether these APEIs are well designed, since they were designed a priori to safeguard this biodiversity and ecosystem function. And this is probably what you would imagine if you, well, you will see later. When you imagine a sponge, when I imagine a sponge, I imagine a sponge like that you can see, you can touch. This is here the Henscheli. It forms sponge rounds. But the one that we're interested in, for comparative purposes, this is the species that I'm interested in this case, which is Plenaster craigie, that it's an endemic species that only grows on top of these manganese nodules. It can be less than 1 centimeter. But it was really important for us to find them in good numbers in there, because it's as an adult, it's sessile, so it will be directly affected by nodal destruction. It's also a filter-feeding organism, which will indirectly be affected by sediment plumes, and it also has licitotrophic larvae with low dispersal ability. So what we did in this case, we collected through different campaigns individuals from one of these protected areas, the APEI 6, and then 3 mining zones. All these samples were collected at around 4,000 meters depth and spanning more or less 1,000 kilometers. In this study, we developed 11 microsatellites, which allowed us to quantify the genetic diversity, which was quite high, and I would say it was significantly higher than for other similar studies for shallow water species, thus conferring this species an unexpected, high expected resilient capacity. But was this genetic diversity shared? This is what we did also, which is we tried to identify which areas were connected or not. And this APEI 6 was connected, kind of connected at least, with 2 of these areas, but there was a clear genetic break, clear genetic separation with this other area, OMS1. And to try to explain this, we developed oceanographic modeling. The particles that you see here, those are random particles that are moving thanks to the predominant current. So it's like the way that the different larvae will get if they were released in there. And as you can see, it's a bit of a mess, but the APEI 6, I mean, these 3 areas are all of them migrating northwards. Thus, there is a mixing of them, while this OMS1, the one in blue in here, is migrating predominantly westwards, and hence the isolation of this area with the rest of the areas. So our data suggested that APEIs, a network, I mean this network, at least for this case in particular, were not well designed. And other APIs should be created to safeguard biodiversity. And this is what we actually achieved some couple of years later, because ISA decided to include 2 new APIs to the south and to the east, which proved that, at least in this case, science can improve management, conservation management, which we are very proud of. Okay, let's now move to the last area I've been working on. It's the North Atlantic. It's in this area, I've been mainly focused on describing what's going on with deep sea sponge grounds, which, as you can see here, those are quite widespread along the North Atlantic. They play a crucial role in this deep sea ecosystem functioning, because they can produce 3-dimensional structures they can behave as nurseries for fish, and they are increasing dramatically the biodiversity as, in a similar way as forests do in terrestrial habitat. They also play an important role contributing to this benthopelagic coupling. So they are important in these biogeochemical cycles in the deep sea. But of course, as it happened with the other two areas that I just mentioned before, there is threatened by humans, and this is, in this case, mainly fish trawling. And this is one of the reasons why they have been protected. They have been recognized as vulnerable marine ecosystems or BMEs, which, as I mentioned, are biodiversity hotspots and ecosystem engineers, but they have a high vulnerability to disturbance, and they also have low recovery potential. This is why they are protected. And this is the work that I've been doing within the past few years through, as a postdoc in London in this European project, Sponges, about sponges. And also thanks to the DeepCom project, which is the Plan Nacional, where I am a PI, and more recently through this Biodiversity Plus project, where I am work package leader. And well, let's go down there to the North Atlantic. This is just to show you how gorgeous and beautiful these areas are, with different species of sponges, different corals. This is the Schultz Massif. I will talk about this later. Other marine invertebrates are in there. These dots that you see here, this is just to give you an idea of how big these animals are. This is 20 centimeters or so in between the 2 points. So these animals can be really, really old. This is the potato sponge, and you can tell why the name of this sponge is Geodegincelli. We'll talk about this later. This is a laser. It's not that they want to kill these species. It's just the lasers to show you how big these animals are. These are hexactinellids also in a rock slope. Other hexactinellids. White corals is not just sponges, but there's some other stuff that they are interesting also. Stoke sponges, as you can see here. And you will see now a vertical wall. This is a vertical wall, and these big, really big animals, those are lithistids, those are sponges also too. Other hexactinellids, so it's full of life. The diversity is huge in there. And if you take a sample, you will find, of course, many other marine invertebrates in there. And some fish, and the fish, you cannot spot them in there. And this is a huge, it's dramatically huge, like 2 meters or so sponge. It can be, I don't know, hundreds of years old. So this is where we're trying to protect. This is what we're trying to preserve with our studies in the North Atlantic. There's also a final part of the video with really beautiful sponges that you will see now. This is also in the slope. You will see the stoke. They are facing the predominant currents. So it's like they are maximizing the filtration rate of the different particles that are in there. Well, I hope I have convinced you that this is really beautiful and we need to preserve this. That was easy. So, but what is that we're doing there in our group? We're using different species. For, in this case, Phacelia ventilarum also forms sponge grounds. It's a sessile organism with alcytotrophic larvae. We managed to collect samples from the Cantabric up to the north of Norway, so quite wide latitudinal range, more than 3,000 kilometers, and a moderate bathymetrical range up to 900 meters depth. And in this case, our genetic connectivity analysis showed that there was a clear separation in between this Cantabric and Roscoff area and the rest of the area, this red cluster. And this red, which means that this red cluster, there's a population, a panmictic population, should be considered as a single population with grouping organisms from Ireland up to the north of Norway. Something that was also corroborated by our migration analysis, in where we didn't find any migration from the southernmost individuals to the northernmost individuals, and we found degree of migration, northwards gene flow among the rest of the areas, especially from Rockall Bank to the rest of the British Islands. And again, in this case, we did oceanographic modeling, and this oceanographic modeling, including the information related to the spawning of these species, corroborated that there was as you can see here, those are the particles that are released through several months, and these particles at the south never reach the particles that are released in the British Islands and the north of Norway, while these ones in here, at the end, appear to be mixing. So it corroborates, again, that oceanographic currents and local topography explains our molecular data. What about genetic diversity? In this case, we found low values for all the different areas, similar to other studies. But interestingly, in the Cantabrian Sea, we found that the genetic diversity was low, was half with respect of the rest of the areas, which also warned us to bear in mind that at least sites for the Cantabrian Sea should be taken into account for protection in the near future due to this limited genetic diversity. But do all species with a similar distribution behave similarly? And this is one of the things that we've been working on lately with my student Carlota and other colleagues from the British Islands with these species. This is Eunithenor vertica. It's a mobile organism, but lives mostly in association with Lopheila petusa. It's a deep sea coral. It's got, well, in this case, we got samples from a similar latitudinal range and a similar latitudinal bathymetrical range. But in this case, the larvae of these organisms is plantotrophic, so it can last in the water column for many different weeks or even months. And what did we found? In this case, we found that both the the traditional markers, the 16S, and also the SNPs, agreed in that there was no separation in between the Cantabrian Sea and the rest, and the British Islands, as opposed to Phacelia ventilabrum. Thus, there was a panmixis between the Cantabrian and the British Islands. So as you can see here, this is the Cantabrian samples that they have the similar genetic pattern, and also the in 16S you have this blue coloring here, which is the Cantabrian samples that are mixed with the other samples. So this puts into light that species with different dispersal abilities might have different genetic connectivity patterns, with the implications for genetic conservation that this might have. And finally, I'm going to talk about what we've done so far with here the Hencheri is an unpublished paper so far. This is the potato sponge that I just mentioned before that appeared in the video. It's got a lysitotrophic larvae. It's got a really huge latitudinal range. We managed to collect samples from across all the whole distribution of the species, from Canada up to the Arctic. But in this case, very importantly, we managed to collect samples deeper than 3,000 meters depth, and this will make a difference, as you will see. This is a spoiler for what's coming. This is, again, collaborative international effort in which we wanted to study both connectivity and adaptation of using both the host and the microbiome, because the microbiome also plays an important role in this species. So this is the results for the neutral SNPs. For the neutral SNPs, we identified two different clusters, orange genetic cluster with samples shallower to 1,300 meters depth, and a deeper cluster with samples in blue with samples deeper than 1,300 meters depth. It means that, for instance, Canada and the Arctic in orange are more closely related genetically than, despite being separated 6,000 kilometers by the fact of being at a similar depth, than samples that were collected in the Schultz Massif, which is a seamount that I will explain in a minute, that were collected in a similar latitude but at different depths. I will explain a little bit better this in a minute. But because I want also to mention that in this case, we also identified different genetic diversity values for shallower and deeper water, which also highlights the importance of quantifying this in order to have, because this might have an effect of the population if the change of the environment occurs. But I would like to come back to what happened in the Schultz Massif in this seamount that I just mentioned before. We found samples with two clear genetic here. Can you hear me now? Yeah. So they were collected in the same spot, but at different depths. As you can see here, this is a seamount. And why is the reason why this is happening? Again, not in this case oceanographic currents, but what we found is that there is a separation of marine water layers that have different salinity, and they are preventing the larvae from deep water and the larvae from shallower water to mix. So there's a barrier that prevents this connectivity to happen. And in this case, and this is just something that we've done quite recently, we wanted to study adaptation to depth in this particular case. So what we did is comparative transcriptomics, comparing organisms from shallower waters and deeper water. And what we found is that there's genes that are differentially expressed, that's a bunch of them, and most of them are related to lipid metabolism, which we believe it's related to the fact that they need to ensure the membrane fluidity, which is different at different depths. Something that has recently been described in a recent study on ctenophores, where they were also finding differences in the lipid content of different the cell membranes of the different species collected at different depths from ctenophores. But I also mentioned before that we were interested in the microbiome, and the microbiome of these species is really interesting because, as you can see here, more than 90% of the tissue is microbes. The arrows just point the cells that are sponge cells. So what we did here also is we, using 16S amplicon sequencing, we characterize the microbiome of these individuals of these species. And although we found, like you can see the same or similar colors, although the microbiome composition of the different individuals was similar, we again found clear differentiation between shallow and deep water species. So in other words, we also found clear separation in between, in Schultz Massif, shallower and deep water samples as we did for the host. So this is for the North Atlantic what we've done so far with other examples of Fakelia species, Hedegenshellia, Enythenor vertica. And this data has already been used by OSPAR to highlight some areas as, to give the status of poor knowledge on some of these areas, and also to start putting some light into areas that should be protected in the near future. So we are also collaborating with OSPAR in this sense. And just to finish, I would like to come back to my initial slide. I would like to think that in the near future, probably in a few years, or I don't know, probably more than a few years, when next time that we type marine conservation, not only polar bears and penguins and stuff like that will appear, but also other potato sponges or stuff like that will also appear, which also are important for conservation in marine habitats. And with that, I would like to thank all the marine and non-marine species that have contributed to this work, and also all the funding agencies, and of course, all of you for your attention. Thanks.
Well, thank you very much for this nice talk and for the beautiful videos. There's time for questions now. So, Pepe.
Bueno, voy a hablar en castellano. El término conectividad es muy amplio, a veces se equipara a dispersión, es decir, la probabilidad de que una larva vaya de un sitio a otro, que sería la conectividad potencial, no hay muchos modelos y tal. Luego está la conectividad real, es decir, que la larva que llegue a un sitio se instale, sobreviva, llegue a estado adulto e incorpore su acervo genético a la población, ¿no? Y eso es suficiente, que haya conectividad genética con que unos pocos ejemplares de cuando en cuando se trasladen de un sitio a otro, garantizan esa conectividad y la diversidad genética. Pero luego está la conectividad demográfica o ecológica, es decir, que el número de larvas que lleguen a un sitio y se incorporen a la población y se reproduzcan sea lo suficiente como para mantener esas poblaciones, ¿no? Las técnicas genéticas son capaces de discernir un poco si existe realmente una conectividad ecológica o demográfica con las técnicas habituales de parentaje, de porcentaje de migrantes y todo esto, que en realidad son cálculos probabilísticos.
Sí, en DeepSea es muy difícil, porque básicamente, para algunas especies es que ni siquiera sabemos cuándo se están reproduciendo. Algunas especies, por ejemplo, Fakelia ventilabrum, que hemos puesto, que teníamos que hemos puesto cuando hacían el spawning, en algunos individuos sí que estaban haciendo spawning, no todos, y no sabemos ni siquiera si es de una manera, se producen ciclos de reproducción anual o si son cuando las condiciones son más favorables. Eso es mucho más difícil de hacer en DeepSea. O, por ejemplo, esta chiquitita esponja, la Plenaster craigie de la Clarion-Clipperton Fracture Zone, no sabemos ni siquiera qué tipo de larva tiene, si tiene larva o no. Entonces, esto para DeepSea es muy complicado. Estudios de este tipo en shallow water, a menos profundidad, sí que se pueden hacer. Sí que se pueden hacer y se pueden cuantificar. Por ejemplo, se pueden recoger, hay algunas especies, por ejemplo, se me ocurre ahora carambe, que tiene una serie de larvas que son fáciles de ver, bueno, fáciles si atinas bien en el momento en el que están produciendo el larval release. Entonces, eso se podía cuantificar cuántas de estas larvas que están ahí acaban siendo luego futuros adultos. Y es decir, y qué porcentaje de estas pueden acabar en otros sitios que están mucho más alejados, porque la realidad es que muchas de estas larvas se asientan muy cerca. No están la semana entera que pueden estar viajando, sino que están allí mismo enseguida. Entonces, esto se podía hacer. Lo que estás diciendo tú a nivel genético sí que se puede hacer, y se puede hacer sobre todo cuando sabes que están liberando los gametos y con especies que son fáciles de recoger, que se conoce la reproducción en especies de poca profundidad.
Que con el tema de las reservas marinas, áreas marinas protegidas en general, hay poblaciones muy fructíferas y que producen muchas larvas y que están exportando a otros sitios, pero esas reservas marinas están recibiendo suficiente número de larvas de otros lugares para mantener esas poblaciones. Esa es una incógnita porque claro, además ahora nos centramos mucho en la conservación, en crear zonas protegidas, ¿no? Pero las larvas que se producen en estas áreas protegidas salen a la corriente marina y si no encuentran hábitats adecuados, bastante repartidos, solo una pequeña proporción de ellas, las que lleguen a otra reserva marina, pues van a sobrevivir. Entonces, si no se conservan hábitats, digamos, parcheados por todos lados, el porcentaje de larvas que, digamos, se desperdician o se mueren es enorme. Bueno, es un tema muy complejo.
Sí.
Y
de ahí que sea, bueno, intento hacer un poco de énfasis en la idea de áreas marinas, de redes de áreas marinas protegidas con corredores que haya, esos corredores serían esas áreas que dices tú que hay intermedias, ¿no? Pues esas áreas también son las que hay que preservar y sobre las que hay que conocer también. Es decir, que habría que estudiar, no solamente poner un foco en ir a muestrear allí y saber cómo se comportan, sino obviamente los alrededores y otras áreas circundantes. Sí, sí, totalmente de acuerdo.
Okay, I switch back to English.
Okay.
Well, thanks for the great talk. There is a long controversy on the origin of the deep fauna, of the deep sea fauna, and the alternative hypotheses are whether these are relics of ancient fauna that's been surviving there, or whether there is always fauna from the upper part of the sea column that goes down and then adapt to the deep sea bottom. That's one part of the controversy. The other one is whether these are endemics of different trenches or the trenches are connected, and then you have kind of metapopulations all around the oceans. So all your studies, are you able to try to distinguish between, or have you test first this, and second, are you able to give a final explanation to this?
On whether this, for instance, for Geodia Gentieli, if they were originated in shallower or deep water, well, I don't know, but probably those are.
And have you made clocks with your
No.
phylogenists to try to understand?
I haven't. We haven't done this, but I guess it could be possible to be done. Yeah, for instance, for Geodia Gentieli, yeah, we may expect this potato sponge. We may expect that these species might have originated in deeper water because this is more where the higher genetic diversity is.
The sister groups are from shallow water or from other places?
They range. They have a bunch of sister species that they are like mostly deeper water species.
Okay, so then maybe you are more in favor of the relic.
But I guess it depends on the species. For instance, the Chondroclide robervalardi, this carnivorous sponge, this can also be found in capes. Well, not this one, but carnivorous sponges, they were originated in deep waters, and they can also be found in capes where they have similar conditions. So, yeah, I guess it depends on. I mean, this Geodia Gentieli, for instance, too, is not occurring in shallower, warm areas. So it's mostly to the Arctic and to the North Atlantic, deep North Atlantic, and then the Arctic, because it cannot stand conditions of warmer areas. So I guess it can live in shallow waters. In
it's
cases.
More about in the past, where they were.
Yeah.
Anyway, just a quick question, very quick. And that he will appreciate as Catalan. How expensive is all this?
You mean what do you mean? The collecting? Really expensive. I think it was. We were collecting in one spot in the Cantabrian Sea with an ROV, and I think it was 20,000 per day. If you multiply this by 20 days or something like that, you can make the numbers. Just one spot, and then you have to go to different places to have different populations to build this genetic connectivity. This is really expensive.
Okay. Hi. I was a bit surprised to hear that, if I'm not wrong, every time you looked into it, you found that there was very low genetic diversity. And recently, I think this is happening across other marine organisms as well. Recently, I heard about Mediterranean limpets. Why? I mean, isn't it a bit strange? Or at least I would have liked to have come to the end of the experiment or the study and be able to say there's not only high species richness, but also very high genetic diversity.
Yeah.
And therefore, it's very important to preserve or something. But instead, the conclusion seems to be there's low genetic diversity.
Yeah. Yeah, I was also surprised about it. And it also depends, in the presentation, I was just trying to put some light into that. It depends on the type of markers that you're using. For instance, for microsatellites, in general, you have like genetic diversity values higher. For SNPs, I don't know why you don't. But of course, we need to increase the number of studies like this to have an idea, a better idea whether this is a general common thing happening across the different species for the SNPs, or whether this is just related to that SNPs are not showing the real genetic diversity. I don't know. But yeah, we need to. I was also surprised, but at least, despite you have like low, you can identify which are even lower, which is also good to spot, to identify which are the most in the areas that can suffer the most in the near future. Yeah, thanks.
And what about population sizes? So did you have a proper sampling to actually recover
Not really.
diversity?
Yeah. For these deep water species, we have a very relatively low amount of specimens, so it's preventing us from doing.
Because that will reduce the measured diversity as compared to the real one to some extent. No? Do you have any more questions?
Hi, you focus mainly on connectivity. This is dispersion. And I was wondering, what about environmental selection? And my question, very particular one, is that if you expect that those species from South Georgia will move to the southern part, polewards, because the temperatures are increasing in the Antarctic Peninsula.
Yeah, that's a good point.
Yeah.
And this is why we need all this whole area to be protected. If we expect that, those individuals in South Georgia are supposed to be more adapted to higher temperatures, and since we were collecting samples from shallow waters, we are expecting that there's going to be a replacement, probably a replacement of individuals. So we're going to be able to spot this in the near future if we continue sampling these organisms, and there's going to be probably a replacement of the haplotypes from South Georgia back to the ones in Western Antarctic Peninsula. That's a good point, yeah.
Yes, thank you.
Thanks.
Any more questions?
Hola, buenas. Gracias por tu charla. Me parece súper interesante. Tengo dos preguntas. La primera es, en este primer trabajo que has presentado, segundo, sobre la zona de esta Cliperion tal, las zonas de protección recomendasteis como añadir 2 zonas al este por lo que habéis detectado en cuanto a la dispersión de la especie. ¿Habéis tenido en cuenta al hacer esa sugerencia que las corrientes que estáis teniendo en cuenta también pueden cambiar, dado que, por el calentamiento global, en plan, no sé si son corrientes a lo mejor muy pequeñas como para verse afectadas por ello, pero... Y la segunda pregunta va más en relación a si sabes si existe un conflicto o algún tipo de piratería en cuanto a las empresas que están ahí minando esos nódulos de magnesio o manganeso, no me acuerdo.
Manganeso, sí.
De manganeso. Porque, no sé, es algo muy común, ¿no? Cada vez que hay un recurso así, además que parece tan escaso, pues enseguida surgen este tipo de asuntos.
En cuanto a las... hicimos una propuesta en este artículo de añadir nuevas en función de los datos, obviamente, que tenemos. No podemos pensar cómo van a cambiar esto, porque además son corrientes muy tenues, a 4.000 metros de profundidad. Es como, tuvimos muchas dificultades. Bueno, no lo hice yo, lo hizo unos colaboradores. Pero, obviamente, de cara al futuro no sabemos cómo puede eso cambiar. Y en cuanto a lo que dices tú de piratería, pues si os habéis dado cuenta en la figura esta que presentaba, el centro es lo único que está minado. No hay ninguna área marina protegida en el centro. Eso me resultó muy raro cuando fui allí de campaña. Y la explicación de eso es que en el centro es donde los nódulos son más grandes. Entonces, en la parte exterior, que es lo que se quiere preservar, es muy pirata. Yo encontraba nódulos que era como, pero si estos nódulos no es los que yo he visto en las revistas, son nódulos chiquititos, algunos un poco más grandes. Pero básicamente la información que tienen las empresas que se van a dedicar a hacer la minería es que la zona esta, que en el futuro, no se sabe cuándo, se va a minar, la optimización sería máxima, porque allí sacan nódulos a reventar. Entonces, sí, hay piratería en este caso, claramente además, desafortunadamente.
Bueno, we're gonna close the talk now, so you can, if you have more questions, you can go to the kiosco with him and with us and ask there, or in the following days. Thank you, Sergi, again for your
Welcome.
talk.